Hello, my name's Martin Sheldon, and I'm going to talk to you about preventing postpartum uterine disease in dairy cattle and how it depends on avoiding, tolerating, and resisting pathogenic bacteria. By the end of the talk, I hope you'll be better able to answer these four questions. How important is postpartum uterine disease? How do cows prevent uterine disease? How do pathogenic bacteria damage the endometrium? And what are the mechanisms providing tolerance to pathogens? Postpartum uterine disease is a common disease of modern dairy cattle. 70 years ago, we had a different breed of dairy cattle that produced very little milk and rarely had uterine disease. Figures are sparse, but less than 9% of animals had any form of uterine disease. The modern dairy cow produces a lot of milk, but about 40% of animals have some form of clinical uterine disease after calving, and more have subclinical disease. For example, this cow has endometritis with pus discharging from the uterus through the vulva and behind this animal. The consequence of uterine disease is lower conception rates, more days open, and increased culling for failure to conceive. Uterine disease is costly as well. Metritis costs farmers up to $410 per case. And endometritis treatment, lost milk production, and replacing infertile animals costs about $2 billion per year between the USA and the EU. Uterine disease is characterized by inflammation and damage in the endometrium. In section A, this is a section of postpartum endometrium, and you can see the lumen is here, the epithelium is here, and then an underlying stroma. And in the expanded section, in section B, you can better see the single columnar epithelial layer and then the underlying stroma. There are areas of damage, and there are areas containing neutrophils, shown by the arrows, which is evidence of inflammation. How do cows prevent uterine disease? Well, an important starting point is that the endometrium is always infected with multiple species of pathogenic bacteria after calving. Whether those pathogens are gram-negatives like E. coli, or the gram-negatives and anaerobes like Prevotella and Fusobacterium, or indeed gram-positives like Trupperella pyogenes and Staphylococcus aureus. These bacteria are often present in multiple interactions. What's interesting in the animals that remain healthy or develop metritis often have a similar microbiome at the start of their process. So after calving on day 0, their microbiome is similar, and it's only when disease develops that we get divergence. So pathogens are always present in the uterus after calving. But not all cows develop disease. Resilient cows prevent postpartum uterine problems by avoiding, tolerating, and resisting pathogenic bacteria. Avoidance is about limiting the exposure to pathogenic bacteria. This might be behaviors to avoid diseased animals, or it might be about behaviors that maintain hygiene. Of course, this is difficult in modern dairy cows because they are kept in intensive conditions, so it is difficult for them to avoid the pathogens and other animals. On the right, in red, resistance is what we mostly think about in terms of disease, and this is about limiting the number of pathogenic bacteria, fighting the pathogens using innate and adaptive immunity to stimulate inflammation and eliminate the pathogens. In the middle is a third concept, and that's about tolerance. Tolerance has been widely studied in plants, but is only more recently being studied in animals. And this is about limiting the damage caused by pathogenic bacteria or excessive inflammation, protecting the tissues against toxins and repairing damage. So it's about tolerating the presence of pathogens rather than killing the pathogens. What we aim for is a robust inflammatory response and rapid repair of damage. So in this cartoon of the first 4 weeks after parturition, I've cartooned resistance in the top section and tolerance in the lower section. What we aim for when the pathogens invade the uterus is a rapid, robust inflammatory response to clear or control the pathogens and then return the endometrium to a normal homeostatic healthy status. Similarly with damage, we aim to have rapid repair and resolution of that damage to return the uterus to a healthy status. Unfortunately, in many modern dairy cows, metabolic stress and tissue trauma is associated with either delayed or inadequate inflammatory responses that result in chronic inflammation and greater damage and less healing that results in chronic pathology and uterine disease. So uterine disease is about compromised resistance or tolerance to the pathogens. In terms of resistance, a great deal of work has been done on that in the uterus, and the main factor is about innate immunity in the postpartum uterus. So that's innate immunity to the pathogens, sensing gram-negative bacteria via their lipopolysaccharide, LPS, or gram-positive bacteria by lipopeptides, which bind to TLR4 for LPS or TLR2 for lipopeptides and drive inflammatory responses in epithelial cells, stromal cells, and immune cells, resulting in the release of cytokines like interleukin-1 and interleukin-6 that control inflammation, and chemokines like interleukin-8 that draw in immune cells like neutrophils that form the pus in the uterus. Pore-forming toxins, which I'll talk a little bit about later, is common from many gram-negative and gram-positive bacteria, and they damage the epithelium and the stroma, leading to pathology. And then that cell death or excess inflammation leads to the release of damage-associated molecules like interleukin-1 alpha, which augments the innate immune response, but in this case to damage. So innate immunity is central to the defense against pathogens and also the response to damage. In terms of tolerance, mechanisms are less clear, but obvious tolerance mechanisms include the epithelium in itself, bound together by tight junctions to form a barrier to the bacteria in the uterine lumen. Mucus on the surface of the epithelium and antimicrobial peptides also help control these bacteria. Of course, the problem in the postpartum uterus is there is epithelial turnover, cell damage, trauma, and infection that exposes the underlying stroma to the pathogens and their virulence factors. So how do pathogenic bacteria damage the endometrium? Well, Trupperella pyogenes secretes a cholesterol-dependent cytolysin called pyolysin that forms pores in cell membranes. And I'm focusing on Trupperella pyogenes particularly because it is the pathogen most associated with severe uterine disease. The fact that it causes cytolysis or pores in cell membranes can be seen on blood agar plates. When you streak the pathogen on a culture plate, you can see there is hemolysis where the bacteria has been streaked. Whereas a deletion mutant that doesn't make pyolysin, when streaked on a blood agar plate, the blood remains red. There's no cytolysis. So T. pyogenes secretes pyolysin as protein monomers. Multiple bacteria and multiple proteins from these bacteria aggregate to form a hole in the surface of the plasma membrane of cells, particularly in cholesterol-rich areas of the plasma membrane. And this leads to the release of ions and proteins through these holes, which ultimately leads to cell death. And this is cartooned more clearly in this slide. So we have the plasma membrane here in these healthy cells, proteins inside the cell like lactate dehydrogenase, which is a protein that's easy to measure, a nice shaped uterus, the actin cytoskeleton, and tight junctions. But if there is damage, bacterial toxins, cell stress, or excess inflammation may cause that damage, and they lead to disruption and blebbing of the plasma membrane. The pores allow the leakage of proteins like lactate dehydrogenase out of the cells. There is barrier breakdown and actin fiber disruption, eventually resulting in lysis of the cell and cell death. Now, this process can be measured. So in here, I'm going to show some endometrial epithelial cells damaged by pyolysine. So on the left, we're measuring the LDH leakage. So here's control. And then here's challenges with increasing amounts of pyolysine causing increasing release and leakage of LDH. Here is cell viability measured by an MTT assay, which is the conversion of a chemical to a color, and is a representation of cell viability. So here's the control challenge, and here's the pyolysine challenge, and increasing amounts of pyolysine increase cell death. And then if you looked at the actin cytoskeleton shown here in white, the nucleus is red, you can see these nice actin fibers and a nice clear outline of the cells. But if you challenge with pyolysine, the fibers are less evident, the nucleus is misshapen, and the arrows are showing or pointing to pores, to disruption, to blebs, and to misshapen cells. What are the mechanisms providing tolerance to pathogens? Well, we've already talked about the epithelium forming a barrier and mucus, but there are other mechanisms. And the mechanism that we've uncovered recently is that oxysterols protect endometrial cells against pore-forming toxins. Oxysterols are oxidized forms of cholesterol. So here's a cholesterol molecule, and in red are a whole series of enzymes that oxidize that cholesterol molecule to a whole range of different oxysterols. And the ones we're interested in are 27-hydroxycholesterol, which is hydroxylated here, and 25-hydroxycholesterol, which is hydroxylated here. And then there are a whole pile of downstream metabolites that you can measure. Oxysterols are important because they regulate plasma membrane cholesterol. So it's not important to understand all the molecular detail, but 90% of cholesterol is in the plasma membrane, and this is derived from LDL cholesterol and from cholesterol biosynthesis. And oxysterols, they limit the cholesterol biosynthesis, they degrade cholesterol to esters, and they increase cholesterol efflux. So increasing oxysterols can reduce cholesterol in plasma membranes. And this is important because oxysterols are present in ovarian follicular fluid and uterine fluid. So if we collect follicular fluid from dominant follicles or growing follicles in the ovary or uterine fluid and use mass spectrometry to identify 25 and 27 hydroxycholesterol, we can find nanogram per mil amounts of 27 hydroxycholesterol and 25 hydroxycholesterol, as well as their metabolites. What's more interesting is that if you take endometrial epithelial cells and you challenge them with virulence factors like LPS, lipopolysaccharide from E. coli, or pyolysine from Truparella, then you have increased release of 25 hydroxycholesterol, whether you measure that as the native 25 hydroxycholesterol or as metabolites, and again in nanogram amounts. If you take epithelial or stromal cells from the endometrium. Here I'm showing the stromal cells, but the principle is the same for epithelial cells. And you treat them with nanogram per mil amounts of 27-hydroxycholesterol before challenge, they are protective. So let's look at LDH leakage. Here's our control challenge, no leakage. Here's vehicle challenge with pyalysine. So the vehicle treatment challenge with pyalysine, you have LDH leakage. But if you treat with nanogram amounts of 27-hydroxycholesterol prior to the pyalysine challenge, then you reduce the LDH leakage. The same with cell viability. Here's our control. Here's the pyalysine challenge of the vehicle treatment, killing cells. And here is protection provided by nanogram per mil amounts of 27-hydroxycholesterol. Here's an image of the cells treated with vehicle and challenge with pyalysine. You can see blebbing, misshapen cells, and loss of the actin cytoskeleton. But if the cells are treated with 27-hydroxycholesterol, when they get a pyalysine challenge, they are better able to maintain their shape and actin cytoskeleton. Interestingly, this is not only limited to cholesterol-dependent cytolysins, but other pore-forming toxins. So Staphylococcus aureus produces an alpha-hemolysin that also forms pores in the plasma membrane of endometrial cells. So here's our vehicle treatment and a control challenge, no LDH leakage. But if we challenge with alpha-hemolysin in the vehicle treatment, we get leakage of LDH. Treating with 27-hydroxycholesterol reduces that alpha-hemolysin-induced LDH leakage. Here's cell viability. All the cells are alive, but killed by alpha-hemolysin. But if you treat with 27-hydroxycholesterol and then challenge with alpha-hemolysin, cell viability is maintained. And again, you see it in the images. These cells are being treated with vehicle, and you can see that the alpha-hemolysin causes cell damage. The cells are all dried up. But if you treat with 27-hydroxycholesterol prior to the alpha-hemolysin challenge, the cells are better able to maintain their shape and size and the actin cytoskeleton. How does this work? Well, 27-hydroxycholesterol reduces plasma membrane cholesterol to prevent the pyalysine binding. So you can detect pyalysine binding by using an anti-pyalysine antibody. So here are two blots. On the left, we've treated the cells with vehicle, 27-hydroxycholesterol and methyl beta cyclodextrin, which is a cholesterol depleting agent, and challenged with a control. And of course, there is no pyalysine. But if you treat with the same things, vehicle, 27-hydroxycholesterol, and the cholesterol depleting agent, methyl beta cyclodextrin, and then challenge with pyalysine, in the vehicle, you have a lot of pyalysine binding, but then 27-hydroxycholesterol and the positive control reduce that cholesterol in the plasma membrane. So the oxysterols reduce plasma membrane cholesterol to prevent binding of pyalysine and to prevent pyalysine damaging the cells. And the quantification of the bands is in the lower graph. So oxysterols, whether they come from follicular fluid or the ovary, or whether they're produced by endometrial epithelial cells in response to LPS or pyalysine itself, the ultimate effect is that these oxysterols help protect cells, particularly stromal cells, against the damage caused by pyalysine released by bacteria like Trupperella pyogenes in the postpartum uterus. And this is an intrinsic protection mechanism that animals can use to tolerate the presence of pathogens. So in summary, postpartum uterine disease is common, it's costly, and it has long-term effects not only on the uterus, but also on the ovaries. Resilient cows prevent uterine disease by avoiding, tolerating, and resisting pathogenic bacteria. In terms of tolerance, our main concern is that pathogenic bacteria secrete pore-forming toxins that damage endometrial cells. And there are several mechanisms to protect cells and allow cows to tolerate pathogens, but the newly discovered route is to use oxysterols that increase the intrinsic protection of endometrial cells against pore-forming toxins and the damage that they cause. So overall, it's important to understand the concept of resilience and to understand you can be resilient to pathogens by avoiding the pathogens, although that's difficult in dairy cows. You can resist the pathogens, and that principally depends on innate immunity in the postpartum uterus. And you can also tolerate the pathogens. You can limit the damage that the pathogens cause. And it's tolerance and resistance that are probably most important in preventing disease in postpartum cows. Of course, we're not hopeless ourselves, and there are a number of things that we can do to perhaps help with resilience in animals. And I'm going to leave you with this last slide, which shows a range of things that we can do as farmers and veterinarians and animal scientists to help cows avoid, tolerate, and resist pathogenic bacteria in terms of managing cows and managing their diet and their vitamins and minerals, avoiding stress and preventing overcrowding and maintaining hygiene, considering using extended lactations, selecting easy calving sires or using female sex semen, maintaining hygiene during parturition and managing dystocia gently, and then identifying and treating animals with uterine disease. In the long term, of course, we can select and breed resilient animals that can themselves avoid, tolerate, and resist pathogenic bacteria.